Hello everyone, my name is Rebecca Sempt and I'm a postdoc in Beth Simone's lab in the imaging platform at the Broad Institute. I'll be co-presenting today with Barbara Diaz-Rohr, who's another postdoc in the lab. Today we'll walk you through some new plugins that are available for CellProfiler to allow you to use Cellpose and StarDist models for segmentation within your CellProfiler pipelines. This workshop is really a passive tutorial that is going to be aimed at experienced users of CellProfiler. So as such, we're not going to be spending time talking about the basics of how to use CellProfiler. We're also not going to be spending time talking about the theory or foundation of image analysis in general. So if you want to learn more about those topics, we strongly recommend that you check out some of our other video tutorials that are available online on the COBA YouTube channel or on our CellProfiler website, or written tutorials that are available on our CellProfiler GitHub repo. So with that out of the way, let's get started with an outline of the tutorial today. First off, we're going to spend a little bit of time talking about what types of images and what types of objects are not suitable for classical segmentation techniques. What about objects or images makes them perform poorly with our classical techniques? And by classical, I mean things like basic thresholding and watershedding or declumping algorithms. And these are cases where we instead may want to use deep learning models to segment our objects of interest. I'll go through a very brief overview of StarDist and Cellpose so you can understand a little bit about the differences between these two. We'll then introduce 3 vignettes that are demo data sets that will demonstrate the performance of these plugins on different types of images. So we'll go through a 3D case with some blastoid images, 2D bacteria images, and 2D neurons in tissue. After that, I'll talk to you a little bit about some considerations for using RunStardist and RunCellpose. Both in terms of how to make them as efficient and as fast as possible, also in terms of which plugin you might want to use for which type of situation. We'll briefly talk about how to train your own model and what resources are available to help you do this. Finally, we'll discuss some resources that are available for further learning and if you have more questions. I should mention that all of the materials that are used today, so the images as well as the pipelines, are available for you to download at this short link, broad.io/cpsd tutorial. So you can download the images and the pipelines there and see a little bit more about how to use these plugins in your own data. So getting started, for many biological image data sets, standard methods for segmentation are perfectly serviceable, and these may be the best option in a lot of those cases. There are some types of images, however, and some types of objects where the standard methods experience difficulties, and this is where deep learning models tend to shine really well. Let's talk a little bit about that. Here are a couple of images from the tutorials that we'll be showing you today from the data sets. I'm going to show each and talk a little bit about why they are challenging. So first, this is an example of some 2D bacteria. And here, this is a challenging case because these objects are irregularly shaped. You can see some of them are kind of long and spindly, and others are shorter. And they're also not round. Many typical or classical methods are built for round objects, whereas these are kind of elongated shapes. Another thing you can see in this image is that there's some degree of clumping and overlap between the objects, which is also challenging for a lot of segmentation algorithms. This is an example image from the 3D blastoid dataset. And in this case, there are a couple of things that are challenging. One is 3D data in general tends to be a little bit more challenging to segment, to have this additional Z dimension. In addition to that, these cells are highly clustered, and being able to draw lines between all of these individual cells is going to be a challenging task for many segmentation algorithms. Finally, this third case is an example of neurons that are present in a tissue. And in this case, we're seeing a couple of things. We have these donut-shaped cells. We also have heterogeneous brightness across our different objects, as well as different levels of background in different tissue sections. And as microscopy is advancing, through techniques like automated, high-throughput, multi-well imaging, super resolution, and others, scientists are producing more and more large 3D datasets and other large datasets of these challenging images that require different methods for segmentation. StarDist and Cellpose are deep learning tools that aim to make this type of challenging segmentation easier. So now I'll go into a brief overview of both StarDist and Cellpose, starting with StarDist. StarDist is a deep learning tool that utilizes star-convex polygons to detect nuclei and other round objects present in your data. So what do I mean by star-convex? I mean these shapes are relatively round, and the star refers to this radial spreading from a central point, and a star-convex shape is one that doesn't pucker in. You could take a point within the cell and find a position where you could draw a line to the entire edge without that line crossing over multiple edges. So these are roundish objects, not puckered in objects like you might see with some types of neurons or other really elongated cell shapes. So from annotated training data, they trained a model to predict radial distances from each point to the edge of the object. They also trained the model to predict object probability, which is kind of like the probability of a particular pixel or point within the object being the center of the object. It's also related to the distance from the center to the edge of the next nearest object. The network tries to predict radial distances and object probabilities for an image, and then performs polygon selection to map these objects and to resolve overlap between objects. This is part of why StarDist works really well in cases where even when you've got clumped nuclei together. So the way it does this is through something called non-maximal suppression, where we keep the object that has the highest probability, and then remove objects that overlap with this object more than a set overlap threshold. So if an object overlaps too much with our kind of key object that has the highest probability of being real, then we toss that object out. Here's an example of the performance of StarDist on an image of nuclei, and you can see it does very well despite heterogeneity in the size of the nuclei, as well as heterogeneity in the brightness, and they're really quite clumped together in this image, but it does well. When using StarDist, the most important thing probably to consider is it is going to assume star-convex shapes. Again, shapes that are roundish, that don't have dramatic puckering in, or like C shapes. In general, we want roundish objects for this, where a point can see the entirety of the edge. So Cellpose is a similar deep learning tool, but a little bit different. It's a generalist algorithm for cellular segmentation. It's built around the idea that there are a lot of different styles of biological image, and the question of, is it possible to develop a generalist algorithm that works well across styles? Cellpose was built from training over 600 various 2D images from these different styles, altogether more than 70,000 objects. These are some examples of the styles of images I was talking about before. We have images of fluorescent cells, images of bright field, other types of images in natural images, like these images of shells over here. By training a model on a lot of these different styles, they were able, and these are examples of kind of ground truth annotations, they were able to arrive at an algorithm that worked very well across all of these situations. There are a couple of key differences to maybe mention for Cellpose. Instead of training a model to detect particular numbers of objects, like this is number 1, this is number 2, the model is actually judged on how well it can predict cell probability. So does this pixel belong to a cell or not, or more generally an object of interest or not? and flow fields. So this image here is an example of flow fields. Each pixel is color coordinated depending on the radial direction that would map it towards the center of the cell. So it's kind of like a color wheel around the cell object. So the model was trained on predicting these. The use of flow fields is part of what allows Cellpose to perform really well even with non-convex objects. So even with very elongated or C-shaped, very puckery cells, Cellpose can perform pretty well. You can also take the nuclear channel into account to improve the segmentation. So we wanted to make it easier to use StarDist and Cellpose in CellProfiler. So we developed a few plugins to bring these tools to CellProfiler. And I really want to shout out Beth and David Sterling, a previous postdoc in the lab, for their work on these plugins. So RunCellpose and RunStardist were created to allow you to use pre-trained models from Cellpose and StarDist as part of your CellProfiler pipeline. To use them, you need to install CellProfiler from source. We also have instructions for this, step-by-step instructions on GitHub for how to use each of these plugins. So briefly, we'll discuss each of these modules and the settings that are present within the plugins. I'll talk about RunCellpose, and then Barbara will take it away for RunStardist and lead us through a couple of vignettes. First, for RunCellpose, this is what the module looks like when you open it and add it to a pipeline. I'll walk you through each of these different options that we have here within the module. First off is detection mode. This is also, you can think about it like this is the model that you are using from Cellpose to segment your objects. This is the pre-trained model. Below that, we have an option for whether or not you'd like to use Omnipose for your mask reconstruction. Omnipose is a slightly different model that can be useful in certain circumstances. Definitely check out their paper for more details about that and information. Below this, we select the input image to be used for segmentation. This could either be cells or nuclei. If you're segmenting cells, you can also supply the nuclei image to Cellpose. And what this will do is improve the quality of your cell segmentation, though it will not also segment your nuclei. So if you want to segment cells and nuclei, you'll need to include two RunCellpose modules. Next up, we have the expected object diameter. This can just be measured as with the measure tool if you open up one of your images and measure some objects. You also could leave it to 0 to have it try to automatically detect this. Below this, we have cell probability threshold and related stitch threshold. These are thresholds for what is called a cell within your image. So to get a better feel for what cell probability means, you can save your probability image, and what this will do is it will pop up next to your image of segmentation to show you what values of probability are associated with keeping a cell versus throwing out pixels and saying they don't belong to a cell. The default for both of these is to be 0, but you can increase them if you'd like. Next up, we have minimum size, which is a minimum size filter to exclude objects that are too small. And after that, flow threshold, which is the maximum error that is allowed during mask creation. It's another threshold for determining what objects are. And just to reiterate, all of these options here are help modules for additional information if you have questions about any of these settings. After that, we have what you'd like to call the output object. In this case, it's being called RunCellpose, and that's what your objects that are segmented will be called. And then a couple of other options down here is whether or not you have a 3D image, and whether or not you want to use averaging of several models together, and whether you want to use the GPU. The GPU will really dramatically improve speed, and we would highly recommend if you have access to a GPU that you use it and configure it to be used by CellProfiler. And with that, I'll turn it away to Barbara to talk about RunStardist and take us through a couple of vignettes.
Hey everyone, I am Barbara Diaz-Rohr, and like Rebecca mentioned, I'm going to walk you through the RunStardist plugin module in CellProfiler, as well as a short demo of how this modules work and some example data, and then 2 examples of difficult to segment images that we have used these modules for. So first of all, we'll walk you through how to use RunStardist. So this is what the module looks like in CellProfiler. It has a few parameters that you need to change to use in your images. The first being the input image, so the name that you gave on the names and types module to your image that you want to use for the segmentation. So for starters, it usually works better on nuclei, and so round objects, and so usually will be your DNA image. This will be the second input will be the model, so the pre-trained model. The RunStardist package already comes with a few pre-trained models, one being the fluorescent nuclei, another one being an H&E staining nuclei that you can use. But you can also use your own trained model here, so just by selecting custom, then it will give you a file path to then point out CellProfiler to where in your computer the model is stored. And Rebecca will go over how to train your own models later in this presentation. The next will be the name for the detected objects, so whatever you want to name your objects as, so nuclei or some other thing that you're segmenting. And another thing will be if you want to tile your images. So if you're working on really big images, this might be useful to reduce the memory usage and avoid the software from crashing. So you can select the number of horizontal as well as vertical tiles. Another things that are not pointed out here is that you also have the option to save the probability images. So this is the probability that's come out from the module, and that's the probability images that is used for the mask creation to detect your object. And also a button to test the GPU. So if you have access to a GPU, you can test it to make sure that it's available and working in CellProfiler. So now I'll go through how to use this modules on CellProfiler. So for this, we're going to use flight data from our examples in our CellProfiler website. So you go to the CellProfiler website and you look at the examples. We have the fly example data, and you can download it from there. So this is the pipeline that already comes with the example data, and we also have a folder with the images, and it comes with a few sample images for segmenting. And just to show you, so I'm going to compare a little bit of what we can do with identify primary objects to identify the nuclei in this type of images, and then compare it to what we can achieve with like RunCellpose and RunStardist. This pipeline has a few crop modules before the identify primary objects, so I'm just going to run through them without showing them. But then the identify primary objects. So some of you might already be familiar with this if you have used CellProfiler before, but similar to what the other plugins that we talked about, they have the input image that you're going to be using. You have the how to name their objects, and then you have a diameter object, a min and max. And this is where you have an average of like what range of size of objects you have, and as well as threshold strategies, and then ways to decomp the object. So if I step through it, you'll get the segmentation for this object, avoiding like things that were too small or things that are detecting the border, touching the border, and you get the segmentation from the nuclei. As you can see, it actually does a pretty good job. Maybe some under segmentation in some areas, but overall, a pretty good segmentation of the nuclei on this type of images. But next, we can compare this to Cellpose. So in Cellpose, like Rebecca mentioned, you have this detection mode. So you have different options to select from. So we're going to use the nuclei because we're detecting nuclei. We're going to use the same input image as the one before to compare. And then here you can change the expected object diameter. So on the one before, you have a range of objects, but in this one, it's just like an average. So I put 20 in this case. The minimum size of the object, and then you can also change the probability threshold, the flow threshold, and this is the name of the output object. In this case, we're just using 2D images, so no 3D images. And on this computer, we don't have a GPU. But as you can see, running Cellpose, even without a GPU on a 2D image, is actually pretty quick. And you can see the segmentation for the nuclei, which is pretty good, even maybe better than identify primary in some of those areas where you have under segmentation before. Just like this, also StarDist, where again, the input image, same one as we used before. The model, for this case, we have that fluorescent nuclei. The objects, which we didn't change, we have RunStardist, and then we save that probability image. And we're not having the images because they're pretty small. So we can run it. And you see again, this is really quick, even without a GPU on small images. So you can use it on just your regular computer. And like you can see, we have pretty good segmentation for the nuclei, even on this. Then you can open your workspace, and we can look at those images. In case I crop blue, we can select it, and we can actually compare the segmentations between the different segmentation strategies. So you can see you do a pretty good job. There's like other things that you can do. You can also like use some of these objects to then add to them or filter them out. But as you can see, Cellpose and StarDist do have an improvement on what we can do with the identified primary objects, even though that one was already pretty good. But it does help you with segmenting, hard to segment images, whether you have like cells that are really close together, or where the lines might not have really sharp edges in between them. So for the first example, we have a bacterial segmentation. So in this case, like regular methods of segmentation did not work because of the really heterogeneous in both size and shape. So you have some that are small, and some are very elongated and large. Some that have like a little ball on one side. So for those like just regular identified primary objects, and segmentation strategies that we used before were not successful. And also we have cells that are overlapped, and overall the bacteria cells can also be really crowded because they grow on top of each other. So for that, we use RunCellpose module. In this case, taking advantage of the new way of mass reconstruction, the Omnipose, because it really it's good for segmenting like those elongated bacteria. So as I said, the pre-trained model that we used was the bacteria Omnipose. The reconstruction method, we use the Omnipose reconstruction method. We use the object diameter. We put here 0, and that might like make no sense, right? Because like the object diameter, but actually when you put 0, the diameter is calculated automatically, which it was really good for us because like, again, the size of the bacteria would change depending on the treatment. And you can adjust this cell probability threshold up and down depending, like once you like when you're on test mode, like I showed before, you can see, okay, well, I'm not getting like all the mass that I want, and you can see on the probability image, you can scroll through it and see the probability scores values in there, and then you can adjust this such probability threshold so that you get a mask and the object detection that you want. Because this bacteria Omnipose actually was trained on face images, we needed to invert our images so that they look like face images, because actually what we have is fluorescent images for this project. So as you can see, the identified primary objects really do good when like we have nice bacteria that are by themselves and they're like round shaped, kind of looking like nuclei. But when you have clusters of bacteria, then you really have under segmentation. But when we did this Omnipose, Cellpose combined with Omnipose, we saw that it really improved our segmentation. When even on those cluster bacteria, you do see like good segmentation, and you can compare it to each other when you see that we have an improved on the segmentation of the bacteria in this case. But it was even more noticeable when we were looking at the images that were elongated, as you can see here. For this elongated bacteria, then identified primary objects just wants to like crop it into little tiny objects that look like nuclei. But when we run Cellpose, even though it has problems sometimes with like touching objects, it does like extremely well compared to what we had at segmenting this elongated bacteria, as you can see in the comparison. And so I can show you again for this pipeline, when we have like the normal bacteria, even the smaller bacteria, it does really good, and the elongated bacteria does an awesome job at segmenting, which really improved our pipeline. So the second example I want to talk to you about are these blastoids. They are 3D images. Why are they challenging to segment with our regular like watershed and declump objects in CellProfiler? Because they have like very heterogeneous size and shape. They can be overlapping, and also we have some, you can see in this image, actually some decrease of the fluorescence intensity towards the end of the C-stack, some bleaching, and also maybe death problems. You can also see in this other comment, you can see the decrease of fluorescence intensity. So for this, we use RunStardist because the first problem was to actually do some nuclei detection on 3D. When we started working on that, actually there was no available 3D model, so we actually had to create our own 3D model, using actually Google Colab, like Rebecca's going to tell you later on the presentation. So what we use is actually made mask and label images based on the watershed, and then just use some annotation tools to then correct those segmentations, and then use those as ground truth for training our own model on this one. You can say, like, once you select the model, you can select where it is on your computer and use that. In this case, this is, and in this case, like I said, using watershed, we had some problem of under segmentation as well as over segmentation of some of the images, and then when we really have like that drop in fluorescence intensity, then we saw that the watershed was not performing as well, and then we missed some of those objects in there. But with the RunStardist, we do see an improvement on it, but we do see like it's really picking up the nuclei within the cell and within the blastoid, and it's not having any problem with that decrease in fluorescence intensity. So next, we'll go to Rebecca, who's going to talk to you about the third example, also show you how to find the instructions to install the plugins, and some ideas of how to train your own model. And thank you very much.
For our third and final example today, we'll look at some neurons in mouse brain sections in 2D. Here we have an example of one image from this set showing the neuron cell bodies in green, and nuclei are present in this image in blue. The goal of this experiment would be to count these cells, these green cells, which you can see are rather clumped together where they're present, and in general are the minority of cells within the actual image. There are many, many more cells within this section that are not fluorescently labeled. For aficionados, the cell type is serotonin neurons. These are labeled by TPH2 or tryptophan hydroxylase 2, the rate-limiting enzyme in serotonin synthesis. So we want to be able to count these serotonin neurons in this brain section. Let's talk a little bit about why this type of image is challenging. Why are these cells challenging to segment? For one, they're rather heterogeneous in brightness. You can see in this image alone, there are some cells like these that are very bright, and others like this, or some of these cells in the background that are considerably dimmer. So that heterogeneity makes it difficult to segment these cells. That's one reason. Another is that, though it may be a little tricky to see, there are actually a lot of TPH2 positive fibers, like dendrites and axons, that are in the same tissue section. You can see these as dots that are present in the image, or sometimes lines coming off of these cells. So they tend to not be quite as bright as the cell bodies, but in some cases they are. We want to ignore those and only get the cell bodies for this experiment. Another reason that I alluded to before is that these cells are donut shaped. And by that, what I mean is that the cytoplasm is fluorescent, but the nucleus is not in this marker. And what this means is that in the center of the cell, there's this dip in fluorescence intensity, and it makes the cells look a little bit like rings or nuclei, or rings or donuts. And those tend to be challenging to segment, and often will be segmented as kind of C shapes rather than round full cell shapes, largely because of this slightly off-center nucleus. Finally, the cells of interest, as I alluded to before, are rare within this section. So we can't go from nuclei out to cells. We have to detect cells first. And they cluster together where they are found, like these cells here along the midline of this section. So to solve this problem, we have an example pipeline using Cellpose. I should also mention that this pipeline file has additional notes in this upper box here for many of these modules to explain what's happening in each one. So if you're interested in more details about what else is happening in this pipeline, definitely check out the pipeline file itself. So to walk you through a couple of the settings here, the first thing you'll notice is the detection mode set to cyto. That model worked very well out of the box for this type of cell. The input image here is a background subtracted version of the TPH2 or the cell marker. This is to remove some of the fibers that are present and avoid situations where the fibers that happen to form, like a ring or something, get called incorrectly as a cell. In this case, we are not supplying the nuclei image as well. The reason for this is because it would actually improve the cell segmentation, but because there are so many nuclei in the image that are not our cells of interest, we would have to perform additional filtering afterwards to get rid of all those nuclei, because they would be carried forward as cells if we included them as a nuclei image. Here for expected object diameter, we set this just based on measuring some cells with the measure tool and CellProfiler. For cell probability threshold, as well as the flow threshold and the stitch threshold, these were all left to their default values, which perform pretty well in this case, so 0 and 0.4. In this particular case, altering these numbers didn't have dramatic effects on improving the segmentation from the out-of-the-box values. We set a minimum size to exclude any fibers that might be called erroneously as cells. We did not need to invert images in this case. This is an option that exists for, say you have bright field images and the model was trained with fluorescent images. It's going to be looking for bright objects on a dark background, and you have the opposite. You may need to invert your image for the model to work correctly, such as using bright field images and having a model that's trained on fluorescence. But that's not the case here, so we can say no. We did not need to save the probability image. This is not a 3D image. I did use averaging and I did use the GPU. The GPU was very important here actually for having an efficient run. Without this, it would take a pretty long time for these images, even though they're not 3D and they're not particularly dramatically huge. The GPU was very important. The GPU memory share for each worker, this will vary depending on your machine. I set this to 0.3 and I think I had 3 workers running. Also in this pipeline is an Identify Primary Objects module, which I optimized to work pretty well for these images. So you can compare and contrast the Identify Primary Objects versus the RunCellpose. So let's do that now. Here's a zoomed in field of view within the section. Identify Primary Objects is shown here in magenta. So what you can see is it performs well with certain types of cells. Cells that are all by themselves, like this cell and this cell up here, and they're not touching other cells, those get detected just fine. Where it falls flat are cells where the nucleus is kind of off to the side, and so we get that C shape, like here and here, or examples where we have clustered cells or clumped cells. We see some cases of under-segmentation, like this and here, and some cases of over-segmentation, where we have a large cell, but the nucleus is again kind of off to the side. So we can see it doesn't really perform super well, definitely not well enough for all of our cells. Let's compare this to how RunCellpose worked. I'm going to show this in green on top, and I will remove the identify primary objects so we can look at just the RunCellpose. So here we can see, in general, RunCellpose performed very well across all of these types of cells. In cases where the cells were crowded, it correctly distinguished them. It did a pretty good job of identifying cells where the nucleus, this dark pit, was kind of off to the side, and this would have been a C kind of shaped cell for identify primary objects. It did much better with those, and it did better at detecting some of these dim cells. So overall, this was a vast improvement with barely tweaked settings from the standard and using the standard cyto model. So that's the end of our vignettes for today. We have a couple more slides on further information and what to do if you need more help. First, let's talk about what to do if you want to train your own model, if these built-in models are not good enough. So training your own model is something that is pretty computationally expensive. If you have access to a GPU in your machine, that's great. If not, there are free options available to you for training models for StarDist and for Cellpose. You can use zero-cost DL4 Google Colab Notebook, which is free and uses Google-provided GPUs to allow you to train your own model with Cellpose or StarDist. Now, in order to do this, you need to provide images and also provide ground truth. So this can be like a label matrix of where the objects are within your image to train the model. This notebook can be run in browser, and then you can put your custom model in your CellProfiler pipeline. Definitely check out this link for their GitHub repo that has excellent documentation as well as video instructions and extensive written out instructions to help you run these notebooks. And this is just an image of what one of their notebooks looks like. Really great. Let's say you need more help than this. Definitely check out our GitHub repos for information on the plugins and to download them. If you need help installing plugins, we have instructions. This is within the same GitHub repo in an instructions folder or accessible via this link. If you need help installing CellProfiler from source, we have a wiki entry at the CellProfiler wiki that provides instructions for each operating system. You do need to install CellProfiler from source in order to be able to use the plugins. And if you've got further help that you need, either installing or on your specific biological question, we encourage you to make a post on image.sc, which is our forum for image analysis experts, novices, to come together and help each other on analyzing biological data and biological images. So that reaches the end of our tutorial for today. And with that, I would just like to thank everyone involved in the labs at the imaging platform. On top, we have the Simini lab, and particularly, again, shout out to Beth and to David Sterling for their work on these plugins. Definitely should thank the labs that created StarDist and Cellpose as well. Of course, we are building on your work. In addition, I would be remiss not to acknowledge the Carpenter Singh lab, our sister lab within the imaging platform that perform a lot of the profiling on imaging data. And let me get myself out of the way real quick. Thank you to our biology collaborators who really drive us forward and encourage us to make new advances and new tools. And thank you also to COBA, the Center for Open Bioimage Analysis, for funding. And with that, thank you so much for listening, and I hope you've learned a bit about plugins available for CellProfiler. See ya!